Hello everyone and welcome on this 20th edition of Science News. Today the story is inspired by yet another article from the Chemical and Engineering News. This time looking at some of the difficulties in the path from making a molecule that is has potentially health benefits towards it actually becoming a drug, a medicine that can be used by humans. There are many steps that need to be taken on this path, and it's a difficult and treacherous path. And one of them is actually, especially if the initial findings are made by a small company, finding someone who will help you with making enough of the compound in good enough quality so that the later stages of the testing for approval and then for production can be made are important parts. And that's what this article in Chemical Engineering News is about, looking at 3 different molecules currently in clinical development. We will have a look at one of them today called squalamine. The story of squalamine, which is an active ingredient of a drug currently developed for Parkinson's disease under the name ENT-01, is developed by a company called ENT-01, and the production side is handled by a division within Pfizer. As you can see on this slide, the story of this molecule of squalamine goes back nearly 30 years into 1990s, where it was discovered by Michael Zasloff. Actually, when Michael Zasloff discovered squalamine, it was as part of work on antimicrobial peptides isolated from African claw frogs in the 1990s. So these were called maginins. And as part of a continuation of this work, they turned their attention to look for similar compounds in other animals, and they picked the dogfish shark. But after performing these isolations, rather than isolating a peptide molecule, they isolated squalamine as this oligoamine derivative of a steroid core. The company, what they have discovered at these points, the point was that squalamine as a positively charged molecule interacts with the negatively charged groups on cell membranes, and it disrupts interactions between cell membranes and positively charged proteins. And that's what has driven the interest in this molecule further. The company has then looked to using this molecule as a cancer therapeutic and as a therapeutic for wet macular degeneration, but those efforts have failed. And ultimately, the interest has later on been resparked with potential of this molecule for treating Parkinson's disease, as it was found that one of the hallmarks of Parkinson's disease is the formations of various aggregates of alpha-synuclein, which actually, as we now know, start at the nerves, the vagus nerve, close to the digestive system, then proceeds towards the brainstem and the brain itself. And given the fact that alpha-synuclein is a positively charged protein, that sparked interest of the people that have worked with squalamine to try whether it could prove useful for this application too. And indeed, squalamine and another molecule isolated from the Squalus acanthias shark, trodoscamine, both have shown their ability to limit and slow down the aggregation and fibrillation of alpha-synuclein, and therefore be able to help in treating or at least slowing down Parkinson's disease. And that is what the company ENT-01 that was founded around the squalamine and its potential to treat Parkinson's disease is trying to achieve. It's actually now moved squalamine as part of the ENT-01 treatment into phase 2b study and preparing for phase 3. As I said, given the fact that ENT-01 is a small company with limited capabilities in terms of producing, this got help from Pfizer, actually from a division, Center 1 of Pfizer, which deals with these research contract and development tasks, and it is this division that's producing the Squalamine for testing and later on perhaps even for distribution. As an example of the contribution from Pfizer, the original Squalamine synthesis from a key precursor took 16 steps. They have already shortened this by 3 and are further improving and streamlining the remaining steps as well. So I hope this story demonstrates one of the aspects of development of molecules from the bench all the way to the bedside. And I hope to see you again next time. Take care.